NJ College of Science, Nadiad, and my colleague Dr. Hardik Bhatti from M.B. Patel Science College, Anand. Today we are here to deliver you an interesting and very important topic of immunology, which are also related to our health, and that is hypersensitive reactions. Commonly, hypersensitive reactions are known as allergic reactions or allergy. Hypersensitive reactions are also known as allergic reactions in the society. So, what is hypersensitive reactions? Hypersensitive reactions are the reactions which are harmful immune responses to an innocuous antigen which leads to tissue injury and dysfunction. Before we start, what is hypersensitivity? We have to understand what is immune response and what is antigen. So, the word immunus coming from the Latin word that means free to exempt or free of burden, which is related to protection against the pathogenic microorganisms. The exact definition of immunity is body's defense mechanism against the pathogenic microorganisms as well as cancerous cells and foreign particles. Immunity provided by our immune systems. Immune system present only in vertebrate animals and it is distributed throughout the body. So it is integrated body system of organs, tissue, cells and cell products. One of the important property of immune system is that it discriminates self antigen from the non-self antigen. That means immune system is one of the best biometric machines present in our body. Now, what is antigen? Antigen are high molecular weight compound. When enter into the body, it is normally foreignic compound. When enter into the body, it induce the immune systems to give response. Now we are thankful to God that every pathogenic microorganisms have one or more antigenic molecules. For example, capsules, cell walls, toxins, viral capsids, flagella all have antigenic property. So on the basis of antigenic property, our immune system discriminate what is self and what is non-self. So in another word, we can say pathogenic microorganisms have fingerprint on the basis of which our immune systems recognize it. There are some non-antigenic molecules which are pollen, egg white, surface molecules of the RBC, serum proteins and cancer cells. So first we discuss about what type of immunities are there. There are two types of immunity. One is specific immunity and another one is non-specific immunity. Non-specific immunity is also called as innate immunity. The immunity which we obtain by birth. It is once again divided into first line defense mechanism and second line defense mechanism. The first line defense mechanism try to prevent the entry of foreign pathogenic microorganisms, while second line defense mechanism try to remove pathogenic microorganisms or antigenic biomolecules from the foreign materials. Now, the specific immune system is the system which we acquired throughout our life. So, when any foreign pathogenic microorganisms or antigen come in contact with our immune system, our immune systems take photograph or fingerprint of these pathogenic organisms and it memorize it. So, whenever the second time any same pathogenic organisms re-enter into the body, it develops specific response. And hypersensitivity reactions are carried out by specific immunity or specific immune systems. The cells involved in immune systems are innate. In case of innate immunity, phagocytic cells, dendritic cells play important role, while in case of specific immune systems, normally B lymphocyte and T lymphocyte plays very important role. Now, there are two branches of specific immune systems. One is cell-mediated immune systems and another one is humoral or antibody-mediated immune systems. The T cells or the T lymphocyte of the blood cells which play very important role in both the case, particularly the T helper cells. When activated by antigen presenting cells, it once again generate cytokines, and these cytokines molecules further activate T C cells into T cytotoxic cells and B cell into plasma cells, as well as they also develop memory cells. So, plasma cells produce antibody and cytotoxic T cells provides immunity power against parasitic antigens or parasitic pathogenic microorganisms. So, the important characteristics of immune systems are they develop specificity, anamnestic response, as well as one of the more important property is it develop memory cells. These memory cells are remain in the body throughout the life. So, whenever the second encounter of the antigen or pathogenic microorganisms with the immune systems, the response is rapid and higher. And because of this knowledge, it led to the development of new field of immunology, what we call prophylaxis. Prophylaxis means prevention is better than the cure, while the same immune system sometimes cause problems in the host, and what type of problems they cause is explained by my colleague Dr. Hardik Bhatti.
Thank you, Ajit bhai. As he said that immune system is working for the host, but number of time it happens that the immune system got furious, it caused damage to the host cells, and this working against protection is really a difficult job for the host to contain or survive. Immune system works against non-self antigens, but sometimes it acts on self antigen, and that is known as autoimmunity. Simultaneously, when it doesn't work, then it is known as immunodeficiency. We all know it as AIDS, for example. Furious, that is when it get exaggerated, overreaction is known as hypersensitivity. So, technically hypersensitivity is an exaggerated immune response that result into the damage of host cells, and usually this is happen in the subsequent time. मतलब એલર્જી અથવા હાઇપરસેન્સિટિવિટી એ એવી પ્રકારની રોગ છે કે જે સામાન્ય રીતે એકવાર અનુભવ થયા પછી ફરીવાર થતો હોય છે. Technically, mast cells are going to generate IgE and that are going to react with the antigens. Simultaneously, the second mechanism that involves hypersensitivity by T cells, and these T cells are going to produce cytokines, and these cytokines are finally going to damage the host cell. Let us see some historical part of this. Climate Perquit in 1906 has coined the word allergy. Allergy means allows, that is something else, other, and argon means work, that immunity is working not for host, for somebody else or something else. The development or the identification of IgE, immunoglobulin, is a type of immunoglobulin that is being detected by Iwasaka in 1960. Usually immunoglobulin E is producing against usually producing against parasitic infection, but this is a non-parasitic type of antigen. When enters immunoglobulin E is also generated, and that is called an allergen. Hans Hinser and Robert Coombs have classified this hypersensitivity in two categories. Number one is depending upon time. One is immediate hypersensitivity, the another is delayed type of hypersensitivity. Immediate means it occurs within short time of period, while delayed is considered as it takes a bit long time. While Coombs in 1963 has evaluated and classified it in a different manner, that which type of immunoglobulin involved. According to it, they have classified it in the 4 category. Type 1 is involving IgE, type 2 involve IgG or IgM type, while type 3 is involving again IgG type, while the delayed type is not involving any antibody, but here the cells are involved, that is, it is cell mediated type, where the T subtype, that is delayed type of hypersensitivity T cell is involved. Let us see what are the causes. મિત્રો આપણે જરા ધ્યાન આપીએ કે આ શેના કારણે થાય છે એ જોવાનો પ્રયત્ન કરીએ. The causes of hypersensitivity or allergy is depending upon number of factors like genetic basis, the food that we eat, even non-food proteins, some toxins, over-conscious of hygiene, and environmental factors that are changing nowadays. Number of allergen has been detected and reported. We can exemplify it as dust, pollen, dander, peanuts, seafood, wasp, ant like creatures, drugs like penicillin, and number of microorganisms are involved in it. To become hypersensitive or to become allergic is depending on a number of factors that also involve our habits. That is, આજકાલના જમાનામાં આપણે જરા ફાસ્ટ ફૂડ, સોફ્ટ ડ્રિંક્સ, પરફ્યુમ્સ, કોસ્મેટિક્સ, સ્મોકિંગ કરવા માટે જે કંઈ કેમિકલ્સ જનરેટ થાય છે એ પણ એલર્જન તરીકે કામ કરી શકે. ચાલો ત્યારે જોઈએ કે જુદી જુદી ટાઈપના હાઇપર સેન્સિટિવિટી છે એ કયા પ્રકારમાં અને કેવી રીતે કામ કરે છે એના મિકેનિઝમ એટલે કે એના પ્રિન્સિપલ સાથે પણ સમજવાનો પ્રયત્ન કરીએ. તો ટાઈપ 1 સમજવા માટે હું અજીતભાઈને રિક્વેસ્ટ કરીશ.
Okay, as explained by Dr. Hardik Bhatti, the classification is purely based on what type of immunoglobulin responsible for the development of hypersensitivity reactions. In case of type 1 hypersensitivity reactions, IgE immunoglobulin as well as mast cell, basophils and allergen play an important role. So what is allergen? Allergen is an harmless non-parasitic antigen which induce production of immunoglobulin IgE into the host, and because of this IgE production, normal IgE production in the human beings is 0.1 to 0.4 microgram per ml, but inappropriate production of serum IgE produce higher amount of IgE. It reaches up to 1 microgram per ml. Now the cells involved in type 1 hypersensitivities are basophil and mast cells. Basophil presents in circulatory systems, while mast cells present in the epithelial surface of the mucous membrane, and it is also present in connective tissue near the nerve cells. These two cells have granules, and within the granules mediators are present. So now we understand how IgE is produced. Generally IgE is produced by a person which we call atopic. It is a genetic defect in which human immune systems produce IgE against non-parasitic antigen, and because of this genetic defect immune systems produce IgE instead of IgG. So as shown in the figure, when allergen enter into the body, antigen presenting cells process it. Here we must understand that the antigen presenting cells present antigen to T helper cell 2, and this T helper cell 2 induce B cells to produce IgE instead of IgG, and also liberates different types of cytokines. This increase in serum IgE level now binds with the surface receptors which we call FcR1 present in or on basophils and mast cell surface. So whenever the antigen second times enter into the human beings or into the sensitized person, it cross-links the two surface receptors present on the mast cell and basophils. So, as shown in the figure, the biochemical events leads to degranulation of the mast cell and basophils. So, how degranulation occurs? When allergen enter into the body second times, in which the mast cells are sensitized by serum IgE. In normal case, serum IgE, the life period, half-life time of the serum IgE is 2 to 4 days. But when it binds to the mast cell surface, it's remain there in a intact form for several weeks. So upon subsequent exposure of the same allergen to the sensitized person, this allergen cross-links the Fc receptor bound IgE molecules. So IgE molecules when cross-linked, two Fc receptor molecules is also cross-links, and within 15 seconds it activate the systems which degranulate the mast cell as well as the basophils. The first response occurs within the 15 seconds, in which protein tyrosine kinase enzymes activated as well as the membrane bound phospholipase C enzyme is also activated. This leads to activate another enzymes, what we call adenylate cyclase enzymes. This adenylate cyclase enzymes convert ATP into cyclic AMP, and the high level of cyclic AMP once again further activate the another protein tyrosine kinase C type of enzymes, what we call protein kinase enzymes. These protein kinase enzymes produce large amount of cytokines. It also produce prostaglandins, as well as it also produce leukotrienes, while at the same time the granules present in the basophils moves toward the outer membrane, and this outer membrane fuse. At that time, the sudden drop of cyclic AMP blasts the basophils, which liberate large number of biomolecules. These biomolecules are divided into two forms, preform as well as the newly synthesized. The preform biomolecules which are liberated during the basophil degranulation, they are generally histamines. Histamines responsible for many different types of problems.